We have completed reverse transcription. Now we will perform second strand synthesis and denaturation. We have 8 molar stock KOH, Qiagen buffer EB, tris, and nuclease-free water. We have second strand reagent, thaw, vortex, spin down briefly, and equilibrate to room temperature. We have second strand primer, thaw, vortex, spin down briefly, and equilibrate to room temperature. We have second strand enzyme on ice, pipette mix, spin down briefly, and place on ice before adding to the mix. Finally, we have slide seals. Remove the slide cassette from the thermocycler adapter and place on a flat, clean work surface. Prepare a thermocycler with the following incubation protocol and start the program. We are ready to denature the captured mRNA. Remove RT master mix from the wells. Ensure accurate dilution of the KOH. Add 75 microliters of 0.08 molar KOH diluted from stock to each well. Incubate 5 minutes at room temperature. Remove KOH from the wells. Add 100 microliters buffer EB to each well. We are ready to prepare the second strand mix on ice. Add the reagents in the order listed in the user guide. We add second strand reagent, second strand primer, and second strand enzyme. Vortex, spin down briefly, and place on ice. Remove buffer EB from the wells. Add 75 microliters second strand mix to each well. Apply slide seal on the slide cassette and place on the thermocycler adapter on the preheated thermocycler. Skip the pre-equilibrate step to initiate second strand synthesis. When the program is done, we will denature the second cDNA strand. At the end of incubation, remove reagents from the wells. Add 100 microliters buffer EB to each well. Remove buffer EB from the wells. Add 35 microliters 0.08 molar KOH diluted from stock to each well. Ensure even coverage. Incubate 10 minutes at room temperature. Add 5 microliters tris to 4 tubes in an 8-tube strip. Transfer 35 microliters sample from each well to a corresponding tube in the 8-tube strip. Do not discard sample. Approximately 1 to 2 microliter volume variation is expected. Vortex, centrifuge briefly, and place on ice. We have completed second strand synthesis and denaturation. Next, we will perform qPCR.